Okay, we'll get started pretty close to 3 o'clock Central Time here. So welcome everyone to this agricultural genome to phenome training workshop. This is on the iPlantSeg Plus tool. I should note that this is the product of one of the AG2PI seed grant awards. And the whole AG2PI project is funded by USDA, NIFA. Just gonna let a few more people in here. So with us today to share this image segmentation tool is Shruti Dashauri. She is a research assistant professor in the School of Natural Resources with courtesy appointment in the School of Computing at the University of Nebraska-Lincoln. She previously was a postdoctoral research associate in the School of Computing at the University of Nebraska-Lincoln and an early career research fellow in the Institute of Advanced Study at the University of Warwick in the UK. She obtained her PhD in computer science engineering from the University of Warwick, a Master of Technology in Computer Science and Application from the University of Calcutta. Her research focuses on computer vision, artificial intelligence, and robotic solutions in smart agriculture and genome to phenome mapping. Also with us is Srinidhi Bashyam. He's a systems software developer in university operations at the University of Nebraska-Lincoln. He obtained his Master's of Science from the School of Computing of the same university, and his research focuses on computer vision, machine learning, and automation, and plant phenotyping. So with that, Shruti, would you like to get us started?
Thank you, Nicole, for the wonderful introduction, and good afternoon, everybody. This is Shruti Das Chaudhuri, working as a research assistant professor, recently being promoted to associate professor at the University of Nebraska-Lincoln. And thank you very much for joining us and for participating in this segmentation techniques and challenges in plant phenotyping workshop. We are going to introduce a newly developed tool named as iPlantSeg Plus, and Srinidhi Bashyam, co-PI of this seed grant, will be is here to demonstrate the tool with me. So the presentation outline, it has two components: theory of segmentation by me and iPlantSeg Plus tool demonstration by Srinidhi Bashyam. Computer vision and artificial intelligence applications in plant phenotyping is gaining momentum over recent years. So some of our newly developed projects as part of the Plant Vision Initiative Research Group have been presented here. So let me initiate the videos. So these are the short videos of different projects from our laboratory. The reason to show about these videos, these short clips, is that all these projects tailored to computer vision and artificial intelligence applications in plant phenotyping, the underlying component of all these things is nothing but segmentation. So segmentation is the fundamental problem in computer vision, and this is our point of consideration today. So some of the projects that have been displayed in this slide, it is worth to point out is that all these projects that I've been showing here, the core component is the segmentation. The fundamental component is the segmentation. So segmentation is the key, and that is what we are going to present today. So the three parameters that play pivotal role in the choice of segmentation algorithms for applications in plant phenotyping are first of all, phenotyping platforms. It can be of two types: controlled environment, that is the greenhouse. Usually we use high throughput plant phenotyping platforms. One of the platforms or facilities are here in the University of Nebraska-Lincoln, that is LemLaTeX Analyzer 3D high throughput plant phenotyping system. And also the field phenotyping. We also have a field-based plant phenotyping platform at UNL, that is SpiderCam. Imaging modalities. The choice of segmentation algorithms also depend on the modalities of images. because visible light, infrared, hyperspectral fluorescent, these are the separate imaging modalities and will require separate computer vision challenges and inquire separate computer vision challenges, and hence will differ in the use of the segmentation techniques. So imaging modalities do play an important role in the choice of segmentation techniques. Another third parameter, which is also very important, is the phenotyping categories. There are two different types of phenotypes, holistic phenotype and component phenotypes. Now, holistic phenotypes consider the whole plant as a single object, and component phenotype considers individual components of the plants like stems, leaves, fruits, flowers, roots, etc. And holistic component phenotypes also play an important role in the choice of segmentation technique, because for computing holistic phenotype, what we only need to see is that to distinguish the plant from the non-plant pixels. So that is kind of easier because we need to segment the entire plant from the background. But in case of component phenotypes, we have to separate individual components of the plants. We have to separate the leaves. We have to separate the stems, the roots, the fruits, and flowers. So separating individual components of the plants require more rigorous segmentation techniques and more challenges more challenges it encounters. Okay, so these are the three main parameters that play pivotal role, and the left-hand side shows the corresponding diagram, a general scheme of segmentation for image-based plant phenotyping analysis, and also we presented the multiple image analysis for segmentation, that is multimodal segmentation, sequence segmentation, and co-segmentation. These are the advanced topics which are beyond the course of this workshop. However, I will cover the traditional and learning-based segmentation techniques in this workshop. And also the evaluation framework, definitely we need to evaluate the quality of segmentation, because the final phenotype that we are going to compute, the accuracy of the phenotypes largely depend on the quality of underlying segmentation techniques. Hence, the evaluation framework consists of quality factor determination, performance evaluation, and we evaluate the performance of the segmentation technique against ground truth. So ground truth is human annotated results that we compare our algorithmic results with. And this is the entire general scheme of segmentation that is finally, or that is used for plant phenotyping applications. Theory of segmentation. Segmentation subdivides an image into its constituent regions or objects. That is, it partitions an image into distinct regions that are meant to correlate strongly with objects or features of interest in the image. Now, an image is a visual representation of a scene that carries a large amount of information. The image captures all objects in the field of view. However, only one or few objects are of interest to us. Separating these objects from the rest is a key computer vision challenge. An object exhibits many common characteristic features that can be used to isolate them. Segmentation, a fundamental problem in computer vision, is the process of partitioning a digital image into multiple homogeneous regions by grouping pixels based on similarity in terms of intensity, texture, and color. Segmentation can also be regarded as a process of grouping pixels together with similar attributes. These attributes could be similar attributes in terms of intensity, it can be texture, it can be color. In image-based plant phenotyping applications, particularly in high-throughput plant phenotyping systems, the objects of interest are often the plant itself or its individual components. It could be stems, leaves, flowers, fruits, and roots for component phenotyping I just discussed. From a practitioner point of view, The three parameters that play pivotal role in the choice of segmentation are the phenotypic platform, imaging modalities, phenotyping categories. The level to which the subdivision is carried depends on the problem being solved. Segmentation should stop when the objects of interest in an application have been isolated. So what are the importance of segmentation? Segmentation is generally the first stage in any attempt to analyze or interpret an image automatically. It bridges the gap between low-level image processing and high-level processing because it's the fundamental problem in computer vision. On top of it, the high-level image processing applications reside. The role of segmentation is crucial in most tasks requiring image analysis, and the accuracy of the task largely depends on the accuracy of the segmentation. The success or failure of the task is often a direct consequence of the success or failure of segmentation. However, a reliable and accurate segmentation of an image is in general very difficult to achieve by purely automatic means. That is why we take into consideration the semi-automatic or the semi-automated techniques, and one of the techniques, a few of the semi-automated techniques will be demonstrated in this workshop today. So a process that partitions the image pixels into non-overlapping regions. Each region is homogeneous, that is uniform in terms of the pixel attributes such as intensity, color, range, texture, etc., and they must be connected. The union of adjacent regions is not homogeneous. So that is the mathematical definition or basis for segmentation. It partitions the image pixels into non-overlapping regions. And the approaches could be thresholding based, age based, or region based. Thresholding base is pretty straightforward. We should have a threshold, and anything image pixel below than threshold will be occupied or will be treated as the object of interest, and the anything not or beyond the threshold must be treated as the object which is of not interest to us or the background. So the object of interest is the foreground, and object that is of no interest to us is referred to as the background. And we determine the foreground and background based on a threshold value, and that is called thresholding based segmentation. Age-based segmentation, it detects and links edge pixels to form a contours, and the part of the scene which is of interest to us is considered as the foreground, and the part of the scene that is not of interest to us is considered to be the background. And region-based use texture or color information of a region to form an image to compute the segmentation. Some of the important facts of image segmentation is all pixels must be assigned to regions. Each pixel must belong to a single region only. Each region must be uniform. Any merged pair of adjacent regions must be non-uniform, and each region must be a connected set of pixels. So these are some important facts of segmentation. And region versus age-based approaches. Region-based methods are more robust because regions cover more pixels than edges, and thus more information is available. When detecting a region, you could, for instance, use texture, which is not easy when dealing with edges. And region growing techniques are also available, like active contours, which give a very good choice of segmentation algorithm when detecting the thin architectures are needed. Age-based methods are preferable if algorithms are usually less complex and these algorithms or these objects have well-defined edges, then only age-based methods work. And the age of a region can often be hard to find because of noise or occlusions. That is why I just mentioned, if the object has a clear predefined edges, then only edge-based algorithms will work. Otherwise, region-based methods are preferred. Combination of results may often be a good idea for a good application or for good segmentation results. Strategies of image segmentation classified into two categories based on two basic properties of intensity values. It could be discontinuity and similarity. So discontinuity-based approach partition an image based on abrupt changes in intensity. And similarity-based approach partition an image based on regions that are similar according to a set of predefined criteria. So these are the two different strategies. We call it discontinuity or similarity. Now we will talk about the segmentation techniques. The simplest type of segmentation is frame differencing segmentation, where we have a background image present. So the figure 1, A shows an original image and B is the background image. Now these images are obtained at the Lemnatics Analyzer 3D high throughput plant phenotyping facility at the University of Nebraska-Lincoln. So frame differencing simply requires the subtraction of the background image from the foreground image or the original image, and the result of operation is shown in the figure C, foreground image. Now, if we closely look at the foreground image, we will find there are some artifacts, some soil components or particles are present. Some part of the background cabinet is also present. We do not need them. We do not want them because those part artifacts are not of the interest. We only need the plant here. So what we have done here, we have superimposed the green pixels on top of it. And then what we generated the figure on D. That means the plant pixels which are green in color are superimposed on the foreground image. Foreground image is the image that we obtained by subtracting the background from the original image. And then when we did green color thresholding, we only obtained the plant pixels, and the unwanted or undesirable parts of the image, the soil component or the background cabinet, are gone. And that is the image we obtained at the figure E, foreground obtained by color-based thresholding. And after binarization, we got the binary image shown in F. So this frame differencing followed by color thresholding is a two-phase segmentation technique. It works great for clear backgrounds like Lemnatics Analyzer high throughput plant phenotyping. As we have seen, the background is pretty clear and it works with Windows setting quite well because it's the simplest type of segmentation, needs very clear background, non-overlapping regions and objects, and it worked fantastically. So for more information, you could refer to our publication where frame differencing followed by color thresholding have been explained in detail. Spectral band difference-based segmentation. This is the segmentation technique, works great for hyperspectral or multispectral image analysis. This technique is useful to create binary mask of the foreground from hyperspectral or multispectral imagery. What we have done here, two bands of specific wavelengths that have significant contrast in intensity are first identified. So in our Lemnatec AG2PI, we do have a hyperspectral chamber. The hyperspectral cameras capture information in a wide range of wavelengths at narrow intervals. So we do have the wavelength range from 546 nanometer to 1700 nanometer, and it captures 243 bands in that wavelength range. So we have first identified two specific wavelengths that have significant contrast in intensity. As far as I remember, they are either at 770 nanometer and 690 nanometer. So they are then enhanced by multiplying a constant factor that is shown C and D. So we multiplied by 2, and what happens, the whiter one or the brighter one, the plant disappears to create a background for us, and the darker one becomes darker, more darker. So they are enhanced by multiplying a constant factor and finally subtracted from each other to isolate the plant pixels, the foreground that we show in C. The enhanced foreground image is then binarized using Otsu's automatic thresholding technique. The technique is really old technique developed by the scientist Otsu in the year 1969 to generate a binary mask for the plant. The binary mask is then used to segment all plant images in all bands of a hyperspectral cube for subsequent analysis. So the technique is very easy. Two bands of specific wavelengths have been identified. Then they are multiplied by 2. The idea behind multiplication is that we just want to create a background by disappearing the plant of the brighter band. And then we are subtracting C from D and we are getting the plant pixels. And then after Otsu's thresholding, we are generating the binary mask. Now, these binary masks have been used to isolate the plant pixels from all bands of the hyperspectral imagery. And this is also published in our previously published paper. And this spectral band difference-based segmentation is a useful technique for hyperspectral or multispectral imagery. Point cloud clustering segmentation. Now, this is an example of component segmentation because we need to identify or separate individual components of the plant. We need to separate the stem, leaves, and the top leaf cluster. So what we have done here, an iterative space carving approach is used for voxel grid reconstruction of the plant. I got a question, what are the benefits of using iPlantSeg tool versus PlantCV or OpenCV? We will come to that point for sure, okay? Point cloud clustering segmentation. So here, basically, we are computing a 3D voxel grid of a plant. And what we are using here, an iterative space carving approach for voxel grid reconstruction of maize plants. The stem is segmented using voxel overlapping consistency check approach. I am going to explain what is voxel overlapping consistency check approach. Before that, I would like to share one important thing. There are 3 main components. One is TLC, top leaf cluster. Top leaf cluster denotes the top part of the plant where the last few newly emerged leaves are not attached to distinct junctions. Instead, multiple incompletely unfolded leaves surround a single junction, occluding each other. So the part of this plant enclosed between the stem and the top horizontal line of the bounding cuboid is the top leaf cluster. We all know what a stem is and individual leaves. So these are the 3 components we are going to separate out or we are going to segment out. Now the stem, the height of the bounding cube enclosing the reconstructed voxel grid is considered as the height of the plant. The cross-section of voxel grid at height 0 denotes the base of the stem. We then proceed along height and consider the next adjacent cross-section at height equal to 1. Now, at this level, we count the total number of cross-sections and measure the areas by the number of constituent voxels. If the number of cross-section at the next level is still 1, we conclude that the cross-section is part of the stem and this is how the stem grows. But what happens if we find the total number of cross-sections more than 1, that is 2? So the cross-section 2, we are finding 2 cross-sections on the upper level. It means there is a presence of leaf attached to a junction. And this holds true for maize plants because we know in maize plants, the leaves are growing using bottom-up approach in alternate opposite fashion. So if there is a presence of cross-section, if the number of cross-section is 2, we conclude presence of a junction with a leaf. Note that this assumption holds true for maize plants, as I just said, because in maize plants, leaves emerge using bottom-up approach using alternate opposite fashion, and a single leaf is attached to a junction. We then compute the area of the overlapping regions of each of these two cross sections with the cross section and the previous level. And the cross section that shows maximum overlapping with the previous level is considered to be the part of the stem. And this iterative process grows on until it reaches the termination condition, that is, until it reaches the TLC. When it reaches the TLC, we are not getting even one or two distinct junctions, rather than we are getting multiple junctions, lot of overlapping areas, and then we conclude that TLC has started because we are not able to get a distinct junction at this point, and we stop iterating, and that part is considered as the stem. Now, when the stem is separated out from the plant, automatically the top leaf cluster and individual leaves are also got separated. Now, at this stage, we compute the nearest neighbor classification for point cloud clustering so that individual leaves form individual clusters, and this is how we are getting or separated out the leaves. So this overlapping consistency check and point cloud clustering to segment individual leaves and the top leaf cluster have been explained in our publication as mentioned in the slide. So please go through the publications for more detailed discussions. Color-based segmentation using k-means clustering. Clustering is an unsupervised classification technique for grouping similar data points. K-means clustering is used to partition a set of data points into a pre-specified number of clusters that minimizes the sum of distances of the data points from their cluster center. In this particular center, the value of k equal to 3, that means we are getting 3 clusters. Now, the original image is shown at the top left-hand side corner, and the object in cluster 1 is shown in the middle. Object in cluster 2 is shown in the left bottom, and objects in cluster 3 is shown at right top. So k equal to 3 means we have 3 clusters, and it is an unsupervised classification technique that group the similar data points. Here the grouping has been done based on color features, and it has a very it basically computes the sum of distances of the data points and compute the distances from the cluster center. This is how it minimizes the distance and form the clusters of the similar points. Histogram-based thresholding. This example shows a segmentation based on histogram, which is also pretty good in the case of images where we do have the very distinct foreground and the background. As we can see that there is a simple telephone which we used long ago, land phone, and then we have a background and the foreground is quite distinctive from each other. So we computed the histogram of the image, and we can see from this particular histogram that it has two distinct peaks. So if we segment or if we separate out the histograms at the correct points, we can get a very good segmentation results. So the original image we can see, and the threshold at 75, as we can see 75 is not a midway between two peaks, and so segmentation results is not good. Now thresholding at 180, that you can look at it bottom right, and this segmentation is also not good because we have lot of white pixels, because this is also not a clear midway between the two peaks. But yes, if you segment it as a threshold, it as 130. 130 falls roughly in the middle of the two peaks, and you can see the left bottom is a very good segmentation because it does the thresholding at 130. 130 is the value that lies between the two peaks, and hence the results of segmentation is quite nice. So this is histogram based thresholding. It first computes the histogram of the image and then find out the middle of the two peaks. Now, if the distinct foreground and background is present in the image, then we should have two different or very clear peaks, and the computing the midway of two peaks is not very difficult, and in that case we can get a very good segmentation result. And that is why we are getting a good approximation at the thresholding at 130, because it falls in the midway between the two peaks. Another example of color-based thresholding. What is the objective here? The objective here, threshold using background colors. So most of the cases we threshold based on the foreground colors. So previously I described methods where we are thresholding based on foreground colors. But yes, of course, we can also threshold based on the background colors. And here is one example for that. The question is remove the blue background from the image. So what is the idea? Idea is multiply or enhance the blue channel and subtract it. So what we are doing here, 1 and 2 are G and B. The red channel, green channel are there, and we are multiplying the blue channel by 2 and subtracting it from the summation of the first two. And this is why we are getting the result on our right hand side. So the blue background has been removed using this particular coding, right? So this is color-based thresholding, but removing the background, not based on the color of the foreground. Active contour segmentation, a very interesting segmentation technique. It is an automatic iterative method where locations in the image are identified based on drawing regions, which we call as a seed mask. Active contours grow or shrink these seed shapes to fill the borders of the region in the image. So accuracy of the segmentation depends on the initial seed mask. So here we have taken an example of an Arabidopsis image. I'm going to demonstrate it for your quick reference using the same image, but the results are obtained using MATLAB image segmenter app of the image segmenter app of MATLAB. So here, I hope everybody can see my screen. So we are going to the app section, and then is my screen visible? Okay, thank you. And then we are going to the image segmenter app. Yes, the image segmenter app is in image processing and computer vision. Right. So we will load the image from my file. This is the Arabidopsis image that we are going to segment, and we are going to separate the foreground from the background. We will draw ROIs. Draw ROIs. Now, assisted freehand is highlighted. Here we must notice that the initial drawing of ROIs definitely plays a very important role in the accuracy of segmentation. But for a quick reference, I will not spend much time on it. Just to show you how active contour works, that is my goal here. The reason I'm showing this MATLAB segmenter because our iPlantSeg Plus tool, which is specifically designed for plant phenotyping analysis, it will first do segmentation and then it will compute the phenotypes. The basic phenotypes basically is based on the MATLAB app. And that is why I thought it's useful to show some of the examples here. So I think that's fine. And Okay, and we can point these as well, since these are individual labs. And then we will click on apply, and then we will close the ROI. And next we will go to active contours. Here is better. Sometimes it depends on how many iterations you want. You have to check the correctness or you have to check the performance. So for this particular example, I increase the iteration, but sometimes you need to decrease the iterations because sometimes it just floods into the background. So you need to decide which iteration. So it's kind of a trial and error method. So it depends on your choice of application. And if you click on evolve, it will evolve. The seed mask will evolve to fill up the borders. You can see they are growing. Regions are growing. So if you have spent more time to draw the seed very carefully, you will get very good segmentation. I use active contours, and the results of segmentation is shown in my slide. I used 300 iterations and 300 iterations, and I did seed masking in all the leaves as far as I remember for the results that I showed in the slide. So I will not compute, like I will not wait till it ends, but this is the process of how the active contours work, and you can see it is growing to fill up the borders. So next, coming back to the slides. So active contour segmentation, accuracy of the segmentation depends on the initial seed mask and also the number of iterations, which is not mentioned here. But yes, definitely number of iterations also play a very important role in the success of active contour segmentation. Texture segmentation using Gabor filters. Now, Gabor filter is a very well-known function, has been used in image segmentation for decades. It has found its application in biometric verification largely, and it is a function which is sensitive to texture with a specified wavelength and orientation. Gabor functions are sensitive to textures, hence it is very effective for texture-based segmentation. Segmentation based on differences in texture between the foreground and background. Now, if you look at the sample image of the dog, we can see that the foreground and background has different textures. In these particular cases where foreground and background has differences in texture, Gabor-based segmentation works very well. So Gabor functions are similar to the receptive field profiles in the mammalian cortical simple cells, and human brains process information in multi-resolution levels, which can be simulated by controlling the scale and the direction parameters in Gabor functions. So Gabor functions are effective for segmenting based on the texture, because these functions have different scales and wavelengths, and we can modify those scales and wavelengths to find out the texture information in an image and can be used to segment based on the texture. So this image, if you see, the original image is shown in the left-hand side, and the middle one is Gabor magnitude image. We computed the scales of the magnitude, and then that is the result of segmentation. Then we superimpose the segmentation results on the original image, and the dog has been segmented out, which is shown at the extreme right-hand side. Color-based segmentation using LAB color space. This you can also find at a MathWorks example. So in this particular example, there are 6 major colors. There are 6 major colors, and the LAB space consists of a brightness layer, chromacity layer A indicating where color falls along the red-green axis, and the layer B indicates where the color falls along the blue-yellow axis. Though the approach will be to choose a small sample region for each color. So sample region for red is shown in this particular image. Similarly, we compute the sample region for each of the 6 colors: background color, red, green, purple, yellow, and magenta. And after that, we use these color markers to classify each pixel to each of the groups. Each pixel is classified using nearest neighboring rule. So color-based segmentation using LAB color space has been taken, this example has been taken from mathworks.com, and the coding is also available in that particular website. Segmentation using superpixels and clustering. So what is a superpixel? Superpixels are the result of perceptual grouping of pixels. Superpixels enable to break an image into a set of structurally meaningful regions. So original image, and then we computed the superpixels. That means we broke the image into set of structurally meaningful regions. We divided the image into set of structurally meaningful regions. And once the image is divided into superpixel regions, we use classification algorithm to classify each region. Here we have used the LAB color space and use k-means clustering to cluster the superpixels based on color features. Now, after the superpixel clustering, the clustering result is shown in the bottom left, and then we used a color map to identify or to visually distinguish different parts of the scene like forest, ice, dry land, and water. We just use a color map to back projection to get a back projection of the segmented results that has been shown here to give a more realistic segmentation which can identify the different segments. That means which is a forest, what is the icy land, what is a dry land, and water. So this is a segmentation using superpixels and clustering. So superpixels are nothing but it is enabled to partition an image into set of structurally meaningful regions, very small regions, and then we basically call it a grid, and then we cluster those grids based on color features. It could be texture features, it could be any features that are distinct from each other, and then basically we just cluster them to get the segmentation results. Graph card segmentation. It is a semi-automatic segmentation technique used to segment an image into foreground and background. So this is another example of our Kana plant that we obtained. The images have been obtained in our high throughput plant phenotyping facility. And I am taking a few minutes to demonstrate the graph card segmentation using MATLAB image segmenter app. And then finally, I would hand over to Bashyam for the rest of the discussion. So let's go to the MATLAB app again. So here we load a different image, the image of a Kana plant. Well, so we got a different plant image. Here I will use this particular image to demonstrate the graph card segmentation using image segmenter app of MATLAB. So let me click on the graph card, and here what we need to do, we have to mark the background and then the foreground. Then basically we will obtain the binary mask of the image. So the mark for background I think we are fine in marking the background here, and then click on the foreground. Sometimes we just zoom out to make the image bigger to facilitate the marking. So it automatically, very nicely, it takes all the leaves in just one go. And now what we need to do, we mark the flower as the foreground as well. Fantastic. So show binary, create mask. Yes. So this is the result of very good segmentation results that we can obtain using graph cut techniques. So now we will go to our presentation. Right? So graph cut segmentation, mark the background and the foreground by drawing lines or polygons to perform this segmentation. So this is the result that I just demonstrated. We got it from the Kana, and it's very easy and intuitive using the MATLAB Segmenter app. Now I will discuss segmentation challenges in plant phenotyping. What are the different challenges that we may encounter? Now, greenhouse-based control environment segmentation differs largely with the field-based segmentation in terms of challenges. Because in field-based segmentation, we get illumination variation, we get the presence of weeds, we get the cloudy versus sunny environment. That means a lot of brightness, contrast, etc. And also the diffuse rains. Those are also the other additional hazards that we can encounter. So the illumination variations and also the complexity in plant architecture. If you look at the bottom figure, we have a maize plant as well as we have a cotton plant. Now for the maize plant, it is easy to segment individual leaves, compute leaf angle, other compute component phenotypes, but for the cotton plants, it's not easy to segment individual leaves. So the complexity in plant architecture also creates challenges. Cluttered background, I will come to that example, but these are the common challenges that we have found out. Color and texture similarity between foreground and background. So I presented a lot of techniques to differentiate based on the color and texture features. Now imagine the case where the color and texture similarity happens between the foreground and background. In that situation, the segmentation becomes really challenging. If the plant is too tiny and the background is huge, like, our coleoptile. Think of a coleoptile at the germination phase. When the coleoptiles are too tiny, it is hard to detect from the soil surface. Self-occlusions, another problems to segment the fruit cell flowers if they are occluded by the leaves. Presence of similar looking weeds. I'm coming to an example where we have maize seedlings as well as the wheat seedlings, and it is really hard to identify and separate out the maize from the wheat. So let us discuss these techniques in detail. So these are some of the images for challenges in image-based plant phenotyping analysis. And then let me go to the color similarity between foreground and background. So this is an image where the weeds are coming out from the maize seedlings, from the ground where maize seedlings are also present. The image contains wheat and maize seedlings. The goal is to design a robot for mechanical and organic wheat management. So this picture has been taken for that purpose only. And the cluttered background and the similarity between the desired and undesirable plants add the challenges here. So this is one example. The presence of wheat really complicates the segmentation process. And the cluttered background, because the background is not uniform here, which will also cause additional challenges to segmentation techniques. Ensemble segmentation using deep learning. Now, what is the solution? If we have very tiny coleoptiles, if we have a huge illumination variation, if we have very cluttered background, how could we obtain this segmentation? Now, segmentations, as I mentioned in the beginning, can be learning based and traditional based. Traditional segmentation techniques often, Definitely, there are many traditional powerful segmentation techniques where we use semi-automated segmentations, as I just showed, demonstrated the graph cut, we demonstrated the active contour. Those are the really helpful segmentation techniques. Apart from that, we can do learning-based or deep neural network-based segmentation techniques. Now, if the segmentation becomes really challenging, if you look at this video here, very tiny coleoptiles are there. Even it is hard to detect with naked human eyes. We can generate, we are showing the mask at the right hand side to show there is indeed presence of coleoptiles, but it is really hard to identify them even with naked eyes. It's because they are so tiny, because the illumination variation is havoc, because the images are taken at 2 minutes interval days and nights. So extreme contrast in illumination variation. Moreover, since these are the germination pots, so we have used some colored stone materials. Plant scientists will know the names very well. So to make that adds challenges and makes the background even more cluttered. So in this particular challenging situation, we often use deep neural networks. And here we have used ensemble segmentation learning model. That means not a particular neural network, but an ensemble of several neural networks we have used for this termination detection. Our publication in Frontiers will have detailed explanation of that, but I'd like to introduce the technique briefly. So that is the ensemble segmentation model. We have raw input images and we have the data sets, staining and testing. We have data augmentation. Basically, the innovation is the ImageNet model that I'm showing here. So basically, it is a testing training model, which is pretty similar and same for all other testing and testing model. But the ImageNet is the invention here, where we have created ensemble segmentation model by augmenting the loss function so that it can adapt to the challenges of segmentation I just mentioned. So here is the block diagram of the ImageNet, a novel ensemble segmentation model tailored to the detection and tracking of living organs in cluttered backgrounds and illumination variation. Basically, what we have done here, we combined Inception P3, VGG19, AC ResNet together by modifying the backbone of the UNet architecture and combined them in a way such that it can detect and track the living organs, very tiny coleoptiles at the time of their emergence in the cluttered background and extreme illumination variation. So this is the ImageNet model, and please go through our publication for detailed explanation. But anyway, the video I have shown how challenging it is to segment the tiny coleoptiles, but deep neural network-based segmentation really enables us to achieve very highly accurate results of segmentation. Now, iPlantSeg Plus tool demo. That tool has been developed as part of our seed grant, and I will now invite Srinidhi Bashyam to demonstrate this software. It is a flexible tool for plant segmentation and compute phenotypes. Now, the tool has been developed using MATLAB Image Segmenter app, but it has been modified in such a way so that we can segment the plants, and also it can automatically compute the phenotypes, and the phenotypes are directly stored in an Excel file. Now, this is the first version of the tool that has been released. We will conduct a survey and we will encourage our workshop participants to participate in the survey to share your feedback so that we can improve our version and come up with the next more user-friendly version. So this is the initial version that we are going to demonstrate today. Thank you so much.
Thank you, Shruti. Oops. Thank you, Shruti, for the detailed explanation on segmentation and its challenges. Let me share my screen before I start on the demo. Okay. So the main reason for development of this tool was to streamline all the process that we have in creating segments. We, as Shruti explained, segmentation is the first fundamental step in the process of analysis of image. Many times for machine learning purposes or even for creating ground truth, depending on the usage, we need to segment like large number of images, maybe like thousands or even in the order of 10,000s, and we are interested only in, we would usually be interested only in like part of the image. Like let's say I'm trying to find out a dry part in a plant, like where then we need to have like a lot of images, different variety, different from different plants. But just that reason, and sometimes we just want the healthy plant, and we need to feed those healthy regions to a machine learning pipeline, so that it learns what a dry region in a plant is. So for all these cases, we need a lot of data sets, a lot of data, a lot of segmented results, and these segmentation is a challenge when it comes to a large data set. The idea for this tool is like to streamline that process where we take advantage of all kinds of segmentation methods available in MATLAB currently. So this tool is based on MATLAB. It requires MATLAB environment, and all the result needs to be available in just one place. And go to the tool. So first thing we have to, sorry. I want to present this slide. Yeah, I need to go. Yeah, that's fine. I'll go to the page where I need. Okay. So this is a really simple tool. First thing, like we can download this tool using like by going to the website plantvision.unl.edu/software. You will find the iPlantSeg Plus link to download the iPlantSeg Plus over there. It is a zip file that gets downloaded. Once that's downloaded, there will be just one file in that, the installation file. And installing that will install an app inside the MATLAB's environment. Once it gets downloaded, it's available. It will be available in the app sections over here. I'm pointing to that particular app that I installed here in my environment. So when we click on this, we would get a simple GUI like this, where there will be just like one or two settings max in order to set up the workspace. So the first thing would be the input folder. Input folder is the place where we as a researcher, we store all the images that we want to analyze on, research on in that particular folder. And that would be the input for all the folders. Once the folder is selected, it will show all the images available in that particular folder. So as we can see, like there is just at this particular folder, we have like these many images present, and we can browse through all these images. Once that's done, the other setting is to set an output folder over here where all the results would be stored. Results of these would mainly be the segmented mask of the image. And also as a bonus, we'll also be storing an Excel sheet which has all the which has most of the phenotypes that are selected by the user. So these phenotypes are listed here. Currently, we compute the plant area, the convexal area, the plant height, the aspect ratio of the plant, and then the solidity of the plant. So these are the things that that gets computed right now. What we are doing is we are harnessing the features available in MATLAB's computer vision toolbox and like image processing toolbox, and we are tapping those features and keeping them all at one places, so that there is no wastage of time switching between these apps and these scripts. The main goal is to save time here when we are working on large amount of images. So once these two things are set, as soon as this is set, we could start the process of segmentation. So to keep it simple, we have two modes of operation where this tool can be run in. One is the batch mode. Excuse me. One is the batch mode. Another one is the interactive mode. So batch mode is where we have just one setting that gets applied for all the images in the workspace in the input folder. So that one setting is currently the thresholder settings right now. To keep it simple, it's just we choose one modality. There are multiple modalities, like Shruti explained how thresholding can be done. It depends on it could have like multiple color spaces. Right now, as a simple start point, we have HSV, hue, saturation, and value, where we have to store. We have to input. We would just have the maximum and minimum values for the hue, saturation value for the regions that we are interested in. Right now, it is set to. Right now, it is set to something else. But if I want to set, if I'm interested in segmenting this particular leaf, I would have to specify what hue, saturation, and value this leaf is in, so that it gets applied and gets applied, and all images in this folder would be segmented. Leaves of those images would be segmented, and we would get the desired result in the output folder. There are multiple ways to get to get these values. One way is just to open MATLAB's color thresholder. So that same image gets opened in the MATLAB's color thresholder app here. So idea is to save time to harness all the features in the MATLAB. So once this is opened, you would select an interested area over here. So that would give us hue, saturation, and value over here. We could be more accurate and spend little more time and get an accurate HSV mask value that is pertaining that would get these segments here. Once this is segmented, you will to know what the hue, saturation, value mask is. Currently, we don't have a way to get these values directly into the app. So what I usually did was to export it to a function. Here, once we do that, we would get hue, saturation, value, min and max. Channel 1 is H, 2 is S, and 3 is V. Min and max value would be available here. We could copy those or transfer those values to the place here. So I'll have written it down. I have noted down what value I want. So I would be just having I'll be punching those values in here. So we could test that, test what happens with this mask. So when the values are input in the background, we do multiple things. As Shruti explained in one of the slides, we do frame differencing, and then we do the color thresholding based on this value, and also we remove all the artifacts that are not connected to a component. So there's a concept of connected components. What we are interested is in the connected components. That is, this leaf is connected to the main stem here, and so is the other leaves. But if there happens to be like another speck or a spot or noise in the background that gets segmented because it qualifies as a green color, those gets deleted in the segment. The main intention behind that is like we think that in most of the cases what we observe that is those are like just noises, and the biggest connected component is that of the plant, and we just retain those for accuracy reasons. So once we get this kind of we settle on a value that works mostly on all the images, we can go back to the batch setting, and then we could segment the selected ones. Once this is segmented, we can go to the output folder and check if it really got segmented and if we have the output over there. So I'll go to the output folder. We can see that there is a folder that gets mirrored. Depending on what folder it is, we store the same folder hierarchy, and it gets. We can see that there's a mask that is stored for that particular image over here. And not only that, we can also go and see what Excel it is, and we can see that there is the phenotypes or the main properties of that particular image or plant are stored in Excel file with the image path. So this Excel is really useful if we want to learn something about, if you want to have a record of all these things, or if you want to do other analysis and compute more complex phenotypes on these based on these basic phenotypes. We can select just a single selected image, or there's an option to segment everything in the folder. So the idea is to like when there are like thousands of images, if we to segment and to create like ground truth or anything, usually we won't do it in one go. We'll probably do it in like multiple days, and when user comes back, we need to continue, the app needs to continue where it left off. So that's one big advantage of this. So I can close this workspace, and I can app, and I can come open it and come back tomorrow, and I could continue my work without affecting the previous work. So that's the reason why these things are like we could skip already segmented mask if it keeps a record of what was segmented before. So if I click on segment all, it starts segmenting. There would be like few files where it might not segment with the actual value, but we can always like come back to that and fix it next day by having a better by having a better threshold settings. If that is not if that is not if that doesn't work, if bad segmentation is mainly for mainly to segment a lot of images at once with minimal settings, that works for most of it. But there would be a requirement where some things would not get that is not enough, where we need an accurate segmentation. So that's where we have another mode in which this app operates, that's in the interactive mode. So this is so this batch is done. We can go back to the output and see what all got segmented. So we have like these many segmented images available, which qualified as the green pixels, and all these are segmented out. So and if we go and see the Excel sheet, we have all the data stored here for that. Great. By there is also like error log. If there's something wrong, you won't go into detail in what this error log is. It is mainly for the debugging purpose or for other things. Yeah, for whenever the app doesn't work properly. So that we need not bother right now, but we can go back and see how interactive mode can help us. So let's see. Let's say oh yeah, and also like we can verify the results in the app itself by scrolling onto it. If there is an output image available, output image is the one that got segmented. If there's available, then it will show it on the right side, just next to the image. So it's like a quick verification of the results. We can sanity we can do a sanity check on on all the images that we did quickly. It's very easy. So it compares like two folders with this two folders. One is the input folder, another one is the output folder, and we can verify the ground truth. So that saves a lot of time. Let's say I want to add yeah, let's say I want to perfect this image for some reasons. I need an accurate segmentation for my purpose. I want to do a perfect segmentation. So in that case, what would I do? I'll just randomly pick one, and maybe let's say I want to accurately segment this. For that, we would go to interactive segmentation. In interactive mode, we just have like 3 buttons here. The first one is like a segment, and it will tap into the segmenter app of the MATLAB. Now this is MATLAB's thing, and most of the people, most of us know about who have worked with MATLAB. They know, and Shruti also explained this in her slide just before how this is done. So just zoom it up. Let's see, we can perfectly segment these. I usually quickly do a graph cut. That's easy because I just mark background and need to come out of this zoom thing.
Click on the zoom, click on zoom. Just
okay, so I'll just mark background quickly, and I think it's fine. Sorry. And then quickly I'll do a foreground. So it's done. I can do other stuffs too. Let's see if the binary is good. Seems to be good. I'm exporting. So good. So the main thing over here while exporting the image is when we export, we have to click on export, and it will ask how would you want to store the masked and final segmentation. It will give you an option to change the variable name. The main thing is not to change anything over here, and then just click OK and close it off. This app will figure out where that images are exported, and it will import it when we click on the import button here. If you see on the right side, we don't have each leaf is like stuck to each other over here. There is no gap. We did a good segmentation there in using the segmenter app. If I import it, that should change. I imported it. You can see now the leaves are all separated. So it all looks good. So this is one use case where we would run the batch on everything, like thousands of images, and then there would be like few which we can correct it using interactive segmentation. And not only that, we can also click on compute once we are fine with the output. You can compute recompute the phenotype, and this recomputed phenotype will be stored in the Excel file, and it gets updated for that particular image. So that's the this is the interactive mode where how it operates, and this is for the brassica plant. So the same thing can be applied for others too. Other kind of objects too. Let's say if I want to do segment flower from the Kana, the plant that Shruti showed. So I change my input folder to that Kana, and it gets we can browse all these images in that. So this is the example of Kana. And we can see that in like last few days, there will be like flower present in that in this data set. So if I want to just get flower from that, if I want to get the regions of flower from the Kana, what I would do is I would on all these images, wherever the flower is present, I would just go to the threshold setting, change the threshold setting such that that the HSV matches that of the flower. So I know this because I already did. I checked what is the pixel values for those flower for those flower. Just to save time, I have a pre-recorded value over here. I would be punching that. And bear with me. And one. So I'm gonna test it. So pretty okay, fine. Of course, I can perfect this if I want to for other images, but I'm just gonna run it on everything. So it gets applied for everything. Wherever these errors is mostly because there is no flower in that day, but wherever it finds a flower, it would probably threshold, and it will get that flower for us in as an output. So it's pretty fast. It wouldn't take that amount of time, just few more seconds, I guess, because there are 2 folders in my dataset here.
Srinidhi, can you take a minute to answer a couple of the questions in that chat?
So as we can see, there are like not much in other plants, but on day 28 onwards or 28th onwards, there are like four flowers over here. I can go ahead. I can see that there are some petals missing in this flower. If that is the case, I can always go to the interactive segmentation mode for that particular image, and just get that thing fixed using interactive thing. So that's super easy. I'll just mark background, and I'll just mark foreground here, create the mask, and export it without changing the variables that the MATLAB offers. Close it and then import it. That would be fixed. So, and I could compute phenotype 2 on that. So that's basically the gist of it. Right now, it's very simple. We have just two modes for that, and in background, we use for batch segmentation, we use our script for segmentation, in the links are in the PPT that was shown, like what things we do for the segmentation. But for interactive, it's left to users. They can choose any of the stuff that is available in the MATLAB segmenter tool. It comes with many things like active contours. Even there is an option to just manually delete and add few pixels or regions of pixel in that. So it really depends on the application and the accuracy that is needed for that. But in all other aspects, is as simple as setting a threshold value, and then clicking on the hit button or the segment button here. It would segment all the images in the folder, and it would place that in in an output that could be shared with other people pretty easily and pretty quickly, and it also can be used for others. So I ran these like the two, or like the flower and everything is stored in the same output file. If we want to run a different experiment, all we need is to just change the input folder to point to that new experiment, and change the output folder to point to a new output experiment, and you would get a fresh and all the results will be stored in a fresh data set, fresh result. And that's as easy as that. Any questions so far? Okay, so does Kamal asks that, does this software work on multiple plants and where plant leaves are overlapping? Can each individual plant be segmented even though they have the same HSV mask? So to answer that question, yes, it does. Not in the batch mode currently. I see where it could I can see where it could fail, because right now, based on the algorithm that runs in the background, it selects the biggest component. Most of the software, most of the images have just one interesting object in the back, in the front, and rest would be the background. So currently that mode is like to get that one plant. So I could see that there is a feature that is that could go in the future app, where we could have like setting for multiple plants, or retain all the connected components, all the connected components or segments that are segmented. So in that case, if there is a feature that could retain, is as simple as passing a flag, I guess, that could be the answer for that mode, where we could have multiple plants segmented. Overlapping leaves, yes, it doesn't matter whether leaves are overlapping, because it's a color, it's a feature of color right now. If those both leaves qualify as color with the same color, probably it will also segment that. It's a good question. Thanks for that. Hope I answered that.
Hi. So what happens?
Yeah. Okay. So does this program require a MATLAB license, or is installing the MATLAB runtime enough? So yes, this would require MATLAB, because we are harnessing we didn't want to reinvent the wheels so far. which is already there. It quickly we use the MATLAB's script for the segmented for interactive segmentation. At least for interactive segmentation, we need MATLAB for that. So that's the question. And if we have many folders, do we need to input each folder individually? The answer to that question is it depends. So if all the folders have like a similar images where you could perform the batch settings for all. So when we click on the batch, it segments everything in the input folder. If that is the requirement, then yes, if all these folders could be placed in one big folder and you could point the workspace to that, it will iteratively get all the folders inside that workspace and all the images inside those folders. It iteratively goes through all those images and picks up and does the segmentation. But if those folders needs a different threshold settings, then probably it makes sense to split those folders and point and do the batch segmentation separately. So how plant height is calculated? That's one question. Plant height is calculated by the bounding box. So there is a convex bounding box is like a simple term, but we calculate the convex hull of the image. The convex hull is like let's say I hope I could draw on this. So imagine all the endpoints of this plant being joined by a polygon over here, segmented one, and the height of that polygon would be called as would be the plant height. So that's how it is calculated. Let's see, next question. Lost my chat window. Okay, so meaningful for plant height is meaningful for answers. So I hope I answered all these questions. I'm going to go back to slide to summarize what we discussed so far, so that we could have more questions if that comes up there.
Sure. Same HSV.
Okay, so I'm going to show like results of fluorescent that we did. So this was the well, on the previous research, we had a lot of images that were kind of challenging to do. This mainly we needed a machine learning algorithm which needed a lot of data from a fluorescent image of a plant. So we had to segment. I'm not sure if this is visible for you guys, but there is an image here, fluorescent image. I will zoom into it, and there will be like there are tiny plants in that image. We had to segment these plant images and have the machine learn which is plant and which area of the plant was healthy and which was not. So that was the problem definition, and that was what we wanted to solve. So in order to do that, first we need to segment the fluorescent image, and we need to segment in such a way that we got all the we choose all the class A type. I would just say that we needed two types of regions. Region A and region B. So we needed region A and region B segmented out of from the plant. So that was the data set. So the big data set, many plants, many things, and we have like tricky images. Like I think that day, these 4 days, camera didn't perform very well or something, and we had these kinds of intermediate things which made it difficult to have one mode of operation. We couldn't just do it in one go. So we needed some kind of flexibility on every image or on some images. So that made this tool really helpful to work. And this was the result that we had. We had done many plants like this, and all these are like properly segmented images, images from the fluorescent data. So it's just the plant, no pot or no framework or no without any background. So this was on the fluorescent modality images. So to summarize, we could download it from this website, and just there will be like just setup involves mostly just two pointing the software to an input folder and an output folder where we get all the artifacts and the results stored. There will be like just two modes of operation. A batch mode where there will be a simple shared settings for all the images, and then there is interactive segmentation where users can tailor the settings for each image and fix the image that requires to be fixed. Threshold settings are like simple right now. It's just HSV min and max that can be retrieved from any app that you would like, or even inspecting the image manually, or we could use the MATLAB's color threshold to get the values. And the phenotypes are stored in Excel sheet in the same output folder with all the 6 phenotypes that are shown right now here are computed for that. So that's the demo. There are a lot of things that could be improved on. We hope we get more when during the tool shop, the training. During the survey, we hope we get more, and we got one or two in the chat here right now that we could use it as a future work. So these are few of the future work that immediate future work that we thought we could add it to the next one, like ability to threshold in many other color spaces, not just HSV, like RGB as well. And sometimes this is a tricky one. Sometimes filling hole is good thing, and sometimes it's a bad thing. Good thing is when there are like noises, when a leaf has a lot of specks or something. That time when we segment a leaf, it's good to fill those holes to get good result. But sometimes it's not, because when leaves overlap, then there would be a gap, which is a genuine gap. It's not part of leaf. We shouldn't fill the hole during that segmentation. So it's a double-edged sword. Filling holes it's not optional right now, but we could set it as an optional. It is a tricky one. It cannot be just a flag, because it depends on the size of the hole and everything. It's a tricky thing. So that's in our radar to handle these as seamlessly as possible. Of course, hyperspectral images are a different thing, which we have ideas how we could include these images also in this tool in future. And multi-class segmentation, like what if I want to segment both red flowers into one folder and green leaves into one, or mix and match? What happens to that? So it's a good thing to have. It saves a lot of time for researchers if we have these kinds of options as well in the tool that could make things easier for many people. The idea of this tool is to just give it to someone without much knowledge on computer vision or the segmentation, and to just do a simple, with a simple click of a button, one could produce these output of segmentation.
Means thing means lives with active control and the to clean it down. That example.
Okay. So is there any question till now? Or okay, there's one question here. For plant area, does the total plant matter containing pixels converted to real area? So no, it's a tricky problem, because it's a pixel area, and in order to convert it to a real area, we need some kind of a scale. The scale needs to be, we haven't thought about it yet. We didn't because it's a problem that needs to be solved in a different way. Not currently, we don't have any plans to include any kind of scale conversion. Scale conversion can be included pretty easily, but in order to set up the scale, it really requires user intervention, because every image is shot at a different scale, and it really depends on the image. So maybe a simple scale could convert. It's just a mathematical formula that could be applied or multiplied, but it really depends on what the scale is. Usually, for any picture that is picked, there would be a object next to it, like a coin, or like any other object, like a scale itself on the image. If that is the case, if we can get cues of the scale in that image, then it's possible to convert it. Else it depends on what user thinks the scale is. If we don't have the cue, I hope that answers that question.
Means
okay, so if there are no questions, I'll just show one more demonstration, like one more folder. This one is of let's do maze. Yeah, this one I'll show the maze one. So maze is different, like that we actually saw this in Shruti's slides. She explained how it could be. So for maze, let's I'll just start with the batch one over here. Most of the things boils down to what HSV we select. HSV selected should be such that it is kind of, it applies to most of the images in data set. It is representative of all the images, so that there is really less work left for us to do in the interactive mode. Which is fine. Something wrong. Let's go with this. Yeah, maybe I punched in a wrong threshold settings or not. Okay. Okay, so it worked for these. Probably for this we need a different one. So let's see. Let's try to fix this. It's a good problem to solve. So I'm gonna fix that. Still working. It's okay. It work. So segment selected. Interactive mode. We'll click on select segment and background. Right. And foreground. So so this is pretty good, but just want to show the active contours thing because we have not a perfect mask. So this could actually help the active contour in these cases. So this I'm not sure if you're able to see, but this is a good one because we had mask everywhere around, but active contour kind of fixes it by growing into or growing out of it. So that's one usage of active contour. Like where we could apply multiple steps to it. So first I loaded it, then I did graph cut, then I did active contour, and over that you could also do more. This is a kind of iterative step, and then I could do a paintbrush where I could erase few things that I don't need. I don't need that, so I could erase that, make the brush much small if I want to, and that would give me a more accurate one. So stuff like this can be done pretty easily. To apply it, finally I could put it. And that should be directly available in the app, like that. So stuff like this can be done pretty easily, where we could, if there is like one image that has gone wrong, we could quickly go and fix it and update the whole workspace, or the data can be updated automatically behind. So that's one advantage of this, where everything is at just one place, and we can change one thing, we can come back and change on the next day, and it will all be changed at the same place. So that's one advantage. So if there are any more questions, I would be happy to answer. I'm handing over the stage back to Shruti right now. Thank you.
So Shruti, I want to return to an earlier question
So
about.
thank you very much for attending our workshop. So we covered the basic theory of segmentation, some segmentation approaches from our publications, from our inventions, from what has been available or what is available in MATLAB, how to use MATLAB image segmenter app or easy segmentation techniques. And finally, we demonstrated our newly developed iPlantSeg Plus tool that is an easy top easy segmentation tool that all of you can freely available. You can download it from our website. It's freely available to everybody, and you can just download it and execute. You do not need any image processing background or understanding of complete mathematical formulae. You can just install the app and start using it as the way you use other MATLAB applications. But one thing to note here, this is our first version of release, so we have a long way to go. We will have room for expansions. We have room to grow here. So I would request your feedback. Please email me about the feedback, or also please copy to Bashyam Srinidhi, who is our system software developer. So please email us with your thoughts, feedback, and your expansions of the software that you'd like to have it and would like to develop a better version of it in near future. So thank you very much for your participation, and hope you find the workshop helpful. And that's it from my side. Thanks a lot.
Thanks, Shruti. Thanks, Trinity. There is one question that I would like to return to. Can you guys hear me?
Yes.
Okay, Anthony,
we are
thank
not
you.
able to hear.
Okay,
Okay, now.
can you hear me now?
Yeah.
Okay, thank you. So there was one question earlier, Shruti, that I want you to come back to if possible. What are the benefits of using iPlantSeg tool versus PlantCV or OpenCV?
Right. So iPlantSeg tool is very intuitive, and the users do not need to understand the underlying complex mathematical formulae or image processing backgrounds. It is intuitive. It has a graphical user interface, and it can directly be used by non-practitioners as well. Most importantly, it is the batch mode or interactive mode in OpenCV or PlantCV. You need to have hardcore programming. You need to have OpenCV programming language and background, and how to incorporate OpenCV in Python or C++ or C programming language. But here, this is absolutely app-based. You can just download or install the app in your MATLAB app library, and you can just start using the app just the way you do other MATLAB programming apps. So this is the graphical user interface-based software platform we are trying to launch for making it available to a wide variety of audiences, participants, and practitioners, and novice programmers as well. And another thing is that it works with multiple image modalities because it involves automatic and semi-automated in addition to batch processing of segmentation. So you have a wide variety of exposures, wide variety of options available at your fingertips. So this is how it differs, because OpenCV, you need to write code for programming and functions, and PlantCV is a great tool indeed. That is true, but again, you need to have a basic underlying programming language and the knowledge of those functions, because I did my entire PhD in OpenCV, so I'm quite familiar with it. That's it. So basically, our target is to reach to a wider audience and get it a GUI-based application. And the batch processing works great with major high throughput plant phenotyping platforms because it works well with our Leibniz analyzer high throughput platforms. And for interactive, you have the option to fix it without fixing underlying functions or going to the programming language, just by looking at the app and modify it with the existing image segmenter app library. So that's it. Thank you.
All right, thank you, Shruti. And there is one more question that I'll just verbally address right now about whether or not this workshop recording will be made available. And yes, usually we have our recordings available by the next day, usually just a few hours after the completion of an event. So if you missed the first part of this workshop, or if you just want to review kind of the different aspects of the platform that they demonstrated, feel free to check back at the website, same address that you used to register for this event, and then you will find the full recording there. And then also the access to the software and data set from this project, because it is funded through the Agricultural Genome to Phenome Initiative, is available on our website under the funded seed grant page. So you can just scroll down to Dr. Dasharary's, sorry, Dasharary. I'm so sorry for
That's
that.
fine.
To her to her project box, and there will be links to her papers that she's published on this project, as well, again, as a website to the platform and the
Yes,
data set.
I'd like to add one thing what Nicholas said. So the sample data set that we demonstrated our tool and also the background information about the segmentation techniques and theory and everything. So we created a sample data set. I am not sure whether it will be useful to the participants, but if it is, and if you'd like to have those images, I will be happy to share with you guys. So please let us know. And even if it is helpful to all of you, the data set that we demonstrated our tool, we are happy to release that as well, in case you find it useful. Okay.
And if
So
there are any more
yeah.
questions, I was going to say we have 10 minutes left. So if anyone has a question, feel free to just unmute yourself and go ahead and ask.
Will this work if there are more than one plants in an image?
I'm sorry, Kamal.
Will it work like if there are more than one plants? Like, for example, for seedlings, we have images where there are multiple seedlings and we are monitoring the growth of the seedlings.
Actually, yes. I probably have noticed I showed an example where there are 9 pods in the image and there are coleoptiles in all those 9 pods, right? For the germination timing detection. So we have a recent publication on coleoptile detection, germination timing, and growth tracking. So if you load that particular image and if you use your interactive mode of segmentation and have a seed using active contourer graph cut, that would work because I tested and it worked. So it does work with multiple multiple segmentation, multiple plants in the same image too, but in interactive mode, not in batch mode. The batch mode is defined basically to take into account of in-house greenhole based settings of high throughput plant phenotyping platforms like Lemnatics Analyzer 3D, what we have, and some other universities or institutes have Phenospecs or some other HTTP platforms. So the HTTP platforms for batch processing, we are targeting the high throughput plant phenotyping platform. But for interactive, that is the reason we have launched the interactive mode as well, because the interactive will take care of all the challenging situations, which is not to fix the errors in batch processing, as well as to take into account of challenging situations of presence of multiple plants in the same image, the extreme variation of illumination, cluttered background, lot of other things, presence of weed and presence of the desirable plant like that. Okay, thank you.
Okay, thank you.
Any more questions, please?
Will this recording be available after this
Yeah,
workshop?
I would be happy for this recording to be available, and just Nicholas said that it will be available from our website. So Srinidhi and I both are absolutely fine and happy for the recording to be available. And we also encourage some questions or feedback or suggestions for improvement, because as I previously mentioned, this is the first version and we have rooms to grow, and we will incorporate your suggestions and we will check whether that is feasible and how best to include those suggestions in our next version. Thank you. Any other question, please?
All right, hearing none. Thank you, everyone, for attending today. Thank you, Trinity and Shruti, for your fantastic presentation and for developing this tool and sharing it with everyone. So with that, I think we'll end this and we'll gain 6 minutes back, right? 6 extra minutes for the day. So, all right, thank you, everyone.
Bye-bye.